Hello everyone and welcome back to our channel where we explore the fascinating world of microbiology. Today's topic is bacterial growth, a process that is critical to the survival and success of bacteria. Bacterial growth is an incredibly complex process that involves many different factors, and we're going to delve into it in more detail today. Understanding bacterial growth. Firstly, let's define bacterial growth. Simply put, it refers to the increase in the number of bacterial cells over time. This process is governed by a number of factors such as temperature, pH, nutrient availability, and oxygen concentration. These factors can either stimulate or inhibit bacterial growth. Nutrient availability. One of the most important factors that influence bacterial growth is nutrient availability. Bacteria require nutrients such as carbon, nitrogen, and phosphorus to grow and reproduce. Without these nutrients, bacterial growth will be limited or completely halted. Bacteria can obtain these nutrients from a variety of sources such as organic matter, inorganic minerals, and other microorganisms. Temperature effects. Temperature also plays a crucial role in bacterial growth. Bacteria have an optimal temperature range at which they can grow and reproduce most efficiently. This optimal range can vary depending on the species of bacteria, but most bacteria grow best between 20 to 45 degrees Celsius. Temperatures outside of this range can either slow down or completely inhibit bacterial growth. Acidity or alkalinity of the growth environment. Another important factor is pH. Bacteria can survive and grow at a wide range of pH levels, but each bacterial species has an optimal pH range at which it can grow best. Acidic conditions, low pH, can inhibit bacterial growth, while alkaline conditions, high pH, can promote bacterial growth. Oxygen requirements. Oxygen concentration is also critical for bacterial growth. Oxygen concentration is also critical for bacterial growth. Some bacteria require oxygen to grow, aerobic bacteria, while others cannot grow in the presence of oxygen, anaerobic bacteria. There are also bacteria that can grow in the absence or presence of oxygen, facultative anaerobes. Bacterial growth phases. Bacterial growth can occur in different phases. The first phase is the lag phase, during which bacteria adapt to their environment and prepare for growth. The second phase is the log phase, during which bacteria grow and reproduce at an exponential rate. The third phase is the stationary phase, during which bacterial growth slows down due to a lack of nutrients or accumulation of waste products. The final phase is the death phase, during which bacteria die off due to a lack of nutrients or other adverse conditions. Measuring bacterial growth. Bacterial growth can be measured using different methods such as plate counts, turbidity measurements, and DNA-based methods. Plate counts involve growing bacteria on nutrient agar plates and counting the number of colonies that form. Turbidity measurements involve measuring the cloudiness of a bacterial culture using a spectrophotometer. DNA-based methods involve amplifying bacterial DNA using polymerase chain reaction, PCR, and detecting the amplified DNA using various techniques such as gel electrophoresis or real-time PCR. Antibiotic selection pressure. Bacterial growth can also be influenced by other factors such as the presence of antibiotics, disinfectants, or other chemicals. Antibiotics can inhibit bacterial growth by targeting specific cellular processes or structures. Disinfectants can kill bacteria by damaging their cell membranes or proteins. Other chemicals such as heavy metals can also inhibit bacterial growth by interfering with cellular processes. Microbial relationship and presence. Bacterial growth can also be influenced by the presence of other microorganisms. For example, some bacteria can form symbiotic relationships with other microorganisms, where both organisms benefit from each other's presence. Other microorganisms such as fungi or viruses can also compete with bacteria for nutrients or inhibit their growth through various mechanisms. Agitation and surface area effects. Bacterial growth can also be influenced by physical factors such as agitation or surface area. Agitation, such as shaking or stirring a bacterial culture, can increase the availability of nutrients and oxygen and promote bacterial growth. Increasing the surface area of a bacterial culture, such as by using a bioreactor, can also increase the rate of bacterial growth. Horizontal gene transfer. Bacterial growth can be affected by genetic factors such as mutations or horizontal gene transfer. Mutations in bacterial DNA can lead to changes in the way bacteria grow and adapt to their environment. Horizontal gene transfer, which involves the transfer of genetic material between bacteria, can also introduce new genes that affect bacterial growth and behavior. Effects of bacterial growth. Bacterial growth is not always harmful. In fact, many bacteria are beneficial to humans and the environment. For example, some bacteria help break down organic matter in soil, while others are used in food fermentation or as probiotics to promote gut health. However, some bacteria can also cause disease and infections, and it's important to understand how they grow and spread in order to prevent and treat these conditions. Manipulating bacterial growth. Bacterial growth can be controlled or manipulated for various purposes. For example, bacteria can be grown in large quantities for industrial or medical purposes, such as producing antibiotics or enzymes. Bacterial growth can also be inhibited or slowed down using various methods, such as pasteurization or sterilization, to prevent spoilage or disease transmission. Summary. In conclusion, bacterial growth is a complex process that is influenced by a number of different factors such as nutrient availability, temperature, pH, and oxygen concentration. Understanding these factors and how they affect bacterial growth is important for a variety of fields, including food safety, healthcare, and environmental management. Like and subscribe. Thank you for watching this video on bacterial growth. We hope you found it informative and interesting. If you enjoyed this video, please consider subscribing to our channel to stay updated on new content related to microbiology and other scientific topics. Also, feel free to leave a comment below if you have any questions or suggestions for future videos. Thank you again for your support, and we look forward to seeing you in our next video.